Hello everyone, welcome to my Strategic YouTube channel. Today I am going to demonstrate the Augustus gene prediction tool. So first we'll go the theory part. So it is a gene prediction tool for the eukaryotic genome, and it also the web server based prediction, and you can also run it locally. So fourth thing you have to remember, this it is a very accurate AB initio program, and it can predict alternative splicing and alternative transcript, and it can also predict the 5' UTR and 3' UTR region, and also can report large number of alternative genes. So here in this video I will demonstrate how to use the Augustus to find out the gene from a FASTA file with the help of the species database. To do that, you have to go Google and type Augustus tool. So here go to this site and you can use the Augustus. So you can go directly to the GitHub site. So you can see in Ubuntu and any Debian system, you can just use this script to install the Augustus. So I have one FASTA file of Saccharomyces cerevisiae made by the Trinity de novo transcriptomic tool, and there is numerous of FASTA file. So I want to check whether it is the gene or not. So to check it, I have to install it, the Augustus tool. So let's just copy this script and run it to install and enter. So you have to write the password. So capital Y, yes. So it might take time. So installation is complete. Let's clear all the things and so sorry, directly you can check Augustus. It's okay, it is I'm coming, just enter. So now you can see the whole usage of this Augustus tool. Let's clear it, and so we have the Saccharomyces cerevisiae Trinity FASTA file, and first we have to check the Augustus species list. For that you have to write -- species and equals to help, and I'm going to just save all the output. For that I'm using this script, and one and break t and just list and enter. So here is the long list. I already saved this list in my working directory. So here you can see it is the identifier, and this is the original species name. So my species is Saccharomyces cerevisiae. So let's check it. There is a 3 variant, you can use any of them. So I'll use this database for the gene prediction. I'm just copying this script. Let's just write the name. So for the prediction, you have to again use Augustus, then species equals to, you have to write the identifier name, this one. Let's paste it here, and your file name, the file name is serifc trinity.fastfile. Then you have to add some other argument like dash dash utr, and first time I want to just off utr portion, and gff3 equals to on, and whatever it will come, I need to save this as an output. So for that use break tee and output. So it is the total script. I think it is fine, and let's enter. Okay, I did a mistake, I think. Okay, so here I just miss one dash. So it is the total script, and let's enter. So it is predicting each of sequence one by one. So here you can see the number of sequence. It is already like 190, 200. So the run is complete. Let's check it. So here is the output. Let's check this output. So here you can see this is a result, the start gene G1, and there is the start gene G2, and you can just copy this file and control find, and you can see 6900 of matches, means they predicted 6900 of gene from your transcript list, maybe some isoform from a single transcript. And if you just remove all the lines started with the hash, you will get the gff3 file from here. So you can use the grep function to remove all this hash file to get the gff3 file. So it is the whole tutorial of Augustus gene prediction tool. If you found this tutorial helpful, please give it a like and leave a comment. For more bioinformatics tutorial, don't forget to subscribe to my channel. Thank you.